Right, going back to the agenda for the meeting. So we'll go over the process of generating MPX data. Then we'll look at a high-level overview of the data analysis pipeline. Then we'll also look at the Pixelator pipeline that we use to process data before we start analysis. Then we'll also dive at all the materials that are available online at our website that can guide you through analysis and be a helper. And then we'll end up with some questions that arise during the talk. Right. So when before starting analysis, we're of course generating data, and in that process, we're basically going from cells and proteins to graphs, which is the native data format for MPX data. The technology is based on antibodies, so of course we start with cells that have proteins on their surface, and the barcoded antibodies that we use, they bind to their respective targets. And then there are two major steps in the protocol that follow. Those are the pixelation A and pixelation B. Those use the main reagent, which is the DNA pixel. And that is a rolling circle amplification product, which binds to the antibodies and to the antibodies that are in close proximity. And in pixelation A step, we basically, by binding to these antibodies that are close by, we're organizing them into very tight neighborhoods. So the whole cell is being covered in these many different. That prepares us for the next step, which is pixelation B. And we use the Pixel B reagent in this case to perform another layer of organization on top of the one that was formed in pixelation A, that groups these discrete neighborhoods into a bigger landscape of neighborhoods that form this comprehensive structure on top of the cell. In this process, in the whole process, we're basically building the amplicon. That is what we sequence, which each amplicon contains the spatial coordinates that are formed during the pixelation A and pixelation B step. So in essence, we're going from single proteins on the surface of each cell. Then we're combining these into tight neighborhoods, and then we're once again combining the neighborhoods into a comprehensive graph that represents an overlay or a map of the surface of the whole cell. This whole process is documented in great detail in our paper in Nature Methods. So feel free to go ahead and read that for a very, very detailed overview of this of the technology. So at the end, we have a 80-plex product, and the data that is delivered after running the assay is threefold. So you get relative abundance for all the 80 markers, and you get that for every single cell in your sample. Then you also get a polarity score, again, for each marker and for each cell. And finally, a colocalization score, and that is a combination of all the possible 80 by 80 combinations for each cell. So then let's start with a high-level overview of the analysis process. We have now, at this point, sequenced our amplicons, and we're ready to begin processing our data. And the first step in this process is running the output through the Pixelator pipeline. That consists of 9 different steps, which I will go over in a little bit. But after it has run through, we have 2 major outputs. So one is we get a QC report for each sample. And then we get a pixel file, which is the basically the main output that is used for downstream analysis. That is a bundle of information which contains the AND data or abundance data. Then you have an edge list, which is contains the building blocks for the graph structures. And then you have polarity scores, all the spatial scores, polarity scores, colocalization scores, and metadata for each sample in each cell within the sample. So the final output is the pixel file again with all the with the abundance, polarity, and colocalization data. To set up the pipeline, it's very straightforward. So there are a few dependencies that need to be set up in advance to be able to install the pipeline orchestrator. In our case, this is Nextflow. Once Nextflow is up and running, which you can do by installing all of these different dependencies and also running this command that is shown to install it. Once it's up and running, you basically need to set up a sample sheet that references the FASTQ files that you wish to process. Then you set up, then you basically input the parameters specific for the for your run and also for the pipeline. And then with a simple call to with this command shown here, you can basically start and kick off the pipeline. And now going back to the different steps that I showed in a few slides back of the pipeline, we have 9 different steps that involve that are involved in processing data. First one is amplicon. So in this step, the pipeline basically creates the amplicon. So reads are being concatenated. Any trimming that is necessary is being performed, and some quality metrics are being logged. The next 2 steps are QC steps. So we perform and record various QC metrics, but basically we're checking for extensively for data integrity and recording the QC metrics. Step 4 and 5 are demux and collapse. And in those 2 steps, we're basically assigning markers to each antibody, to each barcode in each read. And then we're taking care of duplicate reads and importantly, generating the edge list, which in the graph step is being further processed. So this large edge list at this point is being split into into separate smaller components that are to represent different cells. In the annotate step, we refine this. So we basically work with the edge list to filter out components that do not represent cells, and they're probably on the smaller end. So those are being filtered out. And we're also generating our first pixel file. Then in the analysis step, we're computing all the spatial scores, so the polarity score and the colocalization score. And then we're finally ending up with the layout step in which, as the name suggests, we're calculating the 3-dimensional graph layouts that we can use for visualization later on. So at the end of this process, you're left with the 3 layers of data that I've been talking about. You have your abundance for each protein. You have a polarity score and a colocalization score for every possible pair of proteins. And now let's start dissecting these one by one. So first, we will look into the abundance data layer. So abundance we can use as it's being used traditionally to annotate different cell types. So we will have the abundances of all 80 markers. Many of those will represent lineage markers. So you can use that information to basically define our cell population. Then moving on to the spatial data, we have the polarity and the colocalization data, and each of those brings a separate mode spatial mode of characterization of the data. Polarity gives us a peek at whether a certain protein is either polarized into some specific part of the cell, or whether there are a large number of small clusters that are present, or whether that there is no clustering pattern, or the protein is just randomly dispersed. With colocalization, we're moving on from just looking at one protein at a time, but instead look at combinations of proteins. So we look at pairs, and then the data modalities are either segregated, which means that the two proteins are basically split from one another. They're far off from one another, or the data can be, again, random, no significant pattern present within the data. And then the two proteins can get colocalized, which is them being in a very close proximity to each other. So the polarity data is based on the Moran's I statistic, which is a spatial autocorrelation statistic. And as I described, it allows us to see whether there are clustering patterns within the data, or whether there's more of a dispersion or this grid-like pattern that is present. And that is basically representing the negative to the positive side of the statistic. Some example of how MPX data looks when we see significant polarization of a protein. Here I'm presenting CD82, which we observe is polarized after activation, after PHA stimulation of T cells, for example. In this case, we're looking at a CD8 T cell. And we can see here we're showing both the abundance and the polarity data within the same set of samples. So we see that after stimulation with PHA, the abundance of CD82 doesn't change significantly. However, once we focus on the polarity of the same protein, then we see that activated cells are actually very much polarized for this protein. And we have also validated this using microscopy. So on the left-hand side here, we can see one CD8 T cell, and we can see the distribution of the CD8 receptor on the surface of the cell that is more or less uniformly distributed. And in the resting cells, so is CD82. There's no significant pattern of polarization happening. However, after PHA stimulation, the CD8 receptor still remains uniformly distributed. However, this time around, if we look at CD8, then we see these clusters on the surface. And that corresponds to a rather increased polarity score as well. And then we move over to the colocalization scores. And here we're basically looking whether two proteins are more present close to one another more than what we expect by just random chance. The negative end of the colocalization score, again, represent a segregation of the two proteins, and a highly positive score would show us that these proteins are actually very close to each other. So just a few examples of what colocalization data looks like and what we see. This is an example in a resting cell. And in your typical resting cell, you can already see a lot of colocalization events. So there are certain protein complexes that are consists of proteins that are colocalized at steady state, so without any perturbation, such as, for example, the MHC1 complex that consists of HLA, ABC, and beta-2 microglobulin. Those are being part of the same complex. They're always colocalized, and we can see that in our data, which indicates a high colocalization score. We have other complexes such as the VLA1 complex, consisting of CD11A and CD18. Those as well are seen to be colocalized. And then we have also another example with the VLA4 complex consisting of the integrants CD29 and CD49D. Again, showing a high colocalization score. But beyond that, we can also look at these multi-protein domains that are extending again beyond just two proteins. And with the example shown here, we're not showing the names of the protein, but we can see that there are multiple proteins forming this large colocalization signature within that particular sample. So all of these metrics have been documented and described in detail on our website, and you can visit that by going to software.pixelgen.com, where you'll find tutorials that will guide you through data analysis. But you can also find example data sets that if you wish to familiarize yourself with the MPX format and data type even before getting your hands on actual on data of your own, you could do that by just downloading one of these data sets and going through the analysis workflow that we'll describe here. Then you also have a lot of documentation regarding Pixelator and the pipeline itself. So let's now focus on the analysis workflow, and that can be summarized in 5 discrete steps. So first comes data handling, where we basically learn how to read in pixel files. That is the native format that we're going to be working with. And then we also see how to, since oftentimes we have multiple samples, so we'll see how to read multiple pixel files and then aggregate them into objects that we can work with. Then, of course, we need to perform quality control to clean and process our data before we dive into the analysis. Then we go to pre-processing, where we need to transform the data to basically get it ready for the analysis. So there's abundance normalization step, there's dimensionality reduction, clustering, and data integration, all things that are typical of the single cell workflow. And then we importantly learn how to annotate different cells using the abundance data. Then an important and an integral step of the analysis is performing differential comparisons. So comparing either different conditions or different samples within the data, and we can use all 3 layers, abundance, polarity, and colocalization to perform these differential analysis. And finally, a very important part of analysis as well, being able to visualize the results. So the data format that we work with, the graph data, is very amenable to being visualized both in 2D and 3D. So in that portion of analysis, we basically will learn how to do that. In terms of analysis platform, we have libraries available for both Python and R. So on the Python side, we have Pixelator, and on the R side, we have Pixelator R. If you use Python, then you of course take advantage of the ScanPy infrastructure. On the R side, you would use primarily Seurat. Right, and then the first step, as I said, data handling. So having run the pipeline, the Pixelator pipeline, you generate a lot of intermediate data that is not necessarily being output. It can be, you could do so if you wanted to, but the main output would be the pixel files for each of your samples. And as I described, with that you get the different layers. So you get the abundance, you get colocalization and polarity as well. But the first step beyond that, having read your files, is to perform to start the quality control. And the first step in the quality control process is cell calling. So that will be basically filtering out cells, or in this talk we'll refer to cells and graph components interchangeably. So yeah. So in this step, we'll basically be filtering out these cells or graph components that are based on their size, basically. So anything that is too small to be a putative cell will basically filter out. And the way we do that is basically by ranking up all the different cells or components here and looking for the knee of that curve where components start decreasing in size rapidly. Second, after getting rid of the small non-cell components, we deal with aggregates. Those do not form often, but are sometimes present, so we need to take care and filter those out as well. Pixelator generates this or ranks each component based on a tau metric that we've implemented. That is basically showing you the complexity of molecules or antibodies in each cell. So these come pre-labeled. Every single component will be labeled by the Pixelator pipeline. So everything that is either a high complexity or a low complexity aggregate will be already labeled as such and can be filtered. But if one wishes to inspect that data in more detail, of course, that can be done as well. After that, now we've done QC, so we move on to basically the pre-processing. So the first step in that will be the cell annotation. Within Pixelator, we offer two different methods for abundance normalization. Our method of choice is DSB, which stands for denoising by background. But we also offer more traditional ways of normalizing data, such as the centered log ratio. The advantage of DSB, which we've recently switched to using, is that data is being centered at 0. So if one were to visualize or use more traditional flow cytometry sort of approaches for gating cells and annotating them that way, that makes it those values make it much easier and much more amenable to those annotation techniques. The next step is data integration, and data integration is important for multiple reasons that will facilitate our downstream analysis. So it will make it much easier to annotate the data when we have to label every individual cluster that we see present within the data, and then it will also make comparisons between different groups and different conditions much simpler. After having integrated the data, then we can move on to clustering. And to cluster, we basically inspect the variability within all the features or the markers, the protein markers in this case. Then we perform dimensionality reduction, and then we finally use clustering. After having defined clusters, the next step is to basically start annotating, and a gateway to that process is basically trying to compare the abundances of different lineage markers between different clusters, and then assign labels based on the abundance of these markers. That is one approach of annotation. Another approach is, as I said, a more kind of traditional flow cytometry style annotation, but that would require, so it's a gating approach, and that of course would require some prior knowledge of your sample. So if you're running, say, PBMCs, then you know that you would expect to see CD4 T cells, CD8 T cells, B cells, and so on. So with knowledge of what lineage markers those cell populations express, you could compose basically a table for which includes both the positive and the negative markers that are defined for these populations, and automate the annotation that way. But again, you need to be familiar with your sample and know what to expect. So after the data has been annotated and clustered, then we can dive into doing all the differential analysis. And usually the first analysis that we do is abundance. So in this example that I'm showing here, we are comparing a differential abundance analysis between resting and PHA-stimulated T cells. And here we've plotted the results using a volcano plot that is showing us on the right-hand side the proteins that become upregulated after stimulation, and on the right side it's showing, the negative side is showing the ones that are actually upregulated in the resting cells. So we're able to easily visualize the differences between the two conditions using that plot. After performing abundance differential analysis, we dive into exploring the spatial metrics, so the polarity score and the colocalization score. And I'll be illustrating how we run a typical analysis in terms of the spatial metrics with this example here, which is a comparison between resting T cells and T cells that have been cultured on an ICAM1 coated surface in the presence of a chemokine of RANTES. So that the expected result from that experiment is that after the stimulation, after activation, cells usually start adhering to surfaces, and that is mimicking the biological phenomenon where T cells get attached to epithelial cells and try to emigrate to tissues that that show high inflammation. So in that process, T cells tend to form this structure called a uropod, which basically helps them move along the surface that they attach to. And from the literature, we know that these uropod structures, they're characterized by the clustering of a few different proteins. So examples of these are several adhesion proteins such as CD50 or ICAM3, CD162, CD43, CD44, and there's multiple other proteins that cluster within that cellular structure as well. But with this analysis, we'll see if we can sort of verify this by analyzing MPX data that we've generated with this experiment. So the first step of the analysis will be to perform some sort of data exploration. In this case, since we're just working with the polarity scores of 80 proteins, we can visualize basically everything and try to visually inspect and spot any differences that stand out. And in this case, we already can notice these differences. For example, CD8, the CD8 receptor appears to be more polarized in stimulated cells. Then we have other proteins that are also showing a clear difference between the two conditions. But let's go on and formally test for this and perform differential polarity analysis. So let's compare the polarity scores between the resting and the stimulated cells. Once we do that, we readily see that there's some proteins that are very polarized after stimulation, and those include CD37, CD162, CD50. If you recall from a few slides back, all of these are proteins that we see in the uropod. So these, now at this point, we know that these are polarized. it always helps to see the extent of this polarization. Giving a polarity score and comparing median polarity scores, we can see that there is a difference, but in terms of the magnitude of this difference, that doesn't help us much understanding that. What helps though is seeing the extent of the polarization, and we can do that by visualizing every single cell here and looking at how many of these cells are actually being polarized. So if we compare the three markers that we show are different between the resting and the activated cells, we see that in activated cells we have between 13 and 50% polarization for these markers. However, after the stimulation, we see that that increases to 60 to 70%. So that is a dramatic increase in the polarization of these cells. So then, after having looked at the polarization, we can move on and look at different pairs of proteins and how they are co-localizing. And comparing the resting and the stimulated samples, we readily spot the difference as well. So in addition to being polarized, these three markers, CD37, CD50, and CD162, they also co-localize to one another, meaning that they form clusters, but in these clusters, these proteins are also very close to one another, so in very close proximity. And visualizing colocalization data with a volcano plot could be difficult to assimilate. After all, we're getting, with the colocalization data, we're getting thousands and thousands of different possible colocalization pairs of proteins. So having a different way of visualizing that could be helpful. One way to do that would be by using a heat map. And here we can higher colocalization scores are indicated by this deep red in the deep red spectrum. So we can readily observe here centrally in this heat map that again CD50, CD37, CD162 are colocalized to one another. But we also see a huge a pattern of a colocalization signature beyond those three markers. Another thing that we see is on the opposite spectrum, the blue kind of colocalization scores, which are negative, and recall previously show us that proteins are segregating, so moving away from one another. We see a lot of this happening simultaneously, and that makes sense because for proteins to move close to one another, they have to displace others. So this is what we're observing here. And yet another way that could even be easier than on DIs than heat maps is using these colocalization networks here, where in a very clear way we can see that we have a colocalization module here consisting of the three proteins that we've been talking about, CD37, CD50, CD162. But aside from that, we also have this overarching sort of profile or colocalization signature around CD43 and many other proteins that colocalize with it, which is representing this other cluster here in the heat map, aside from the three proteins CD37, CD50, and CD162. So then, having these spatial scores, we can plug them into multiple data processing pipelines, such as dimensionality reduction. So one way to use colocalization scores, for example, would be to perform dimensionality reduction and plot them on a UMAP. And by doing that, we can, instead of traditionally, as we used to looking at abundance data on these UMAPs, we can look at the spatial data and sort of get an insight on how the spatial organization of proteins on the cell surface can contribute to their phenotype instead of abundance. So instead of defining these populations that we're used to working with, such as different subsets of T cells, we can use spatial organization of proteins to define different subsets. And in this case, we can see that the resting and the stimulated cells have form these discrete and separate clusters based on the colocalization scores and based on the changes that happen or occur through along the stimulation process. So what we see here is that the resting cells are basically splitting off from the stimulated cells, which we know at this point form these uropods to be able to attach to surfaces. And then we usually end up the analysis pipeline by visualizing cells. Once again, working with graph data, we can leverage this to form these beautiful visualizations of every single cell, in fact, in our data. And in this example, we're just showing the colocalization here between CD162 and CD50 that markers that we looked at previously. As you can see, we've highlighted the places in the cells where these markers, these proteins are present, and you can see that these correspond between CD162 and CD50. And yet another way is to form these graphs as well and to look at them from all angles. So here we're looking at two different cells. The one on the left is a uropod forming cell, a stimulated cell, and we've highlighted the abundance. So the nodes in this graph where the uropod proteins are present. And as you can see, they form this distinct partition or part of the cell here that would represent the uropod. On the right, we have shown a non-stimulated cell from the control group. And as you can see, the same proteins have been highlighted there, and they're more or less uniformly distributed across the cell surface, not forming any significant patterns. So with that, I think I'd like to thank you for listening and open it up for any questions that we might have.